and we'll get started. So hello everyone, and welcome back to the 2021 Small Farms Winter Webinar Series hosted by the University of Illinois Extension's Local Food Systems and Small Farms team. This is Zach Grant once again, today's moderator and webinar coordinator. I'm a local food small farm educator in Cook County, and we're going to take our weekly dive into all things small farm. So we really appreciate you joining us for these webinars, and we're going to do our best to begin and end within this hour. This is a tight time frame, and we're going to get a lot of great info today and learn about Casey's work. So if you want to put your questions in the chat box, I'll go ahead and curate those, and we'll likely take those at the end again. This presentation is being recorded, and I'll email you a link to the archived presentation as usual towards the end of this evening or early tomorrow morning. There will also be a short online evaluation. I'll have the anonymous link in the chat box, and you should see a QR code on Dr. Athid's last slide as well, and you can use that QR code with your phone if you so choose. We really look for your insight with our evaluations. And there is a couple extra questions on there that might help Dr. Athid's research in the future. So definitely want you to fill those out. We're using Zoom again. Haven't had any problems so far, so we're really happy with Zoom. A couple of things to note. We do have a poll, so I'm going to go ahead and launch this poll, and this is just a voluntary demographic poll that we like to use for our extension programming. So go ahead and fill that out when you get the chance. And again, once again, definitely put your questions in the chat box, and I will curate those and maybe answer a few of them if I can as quick responses, but we'll definitely have some time at the end for Q&A with today's presenter. So with that, this week's presentation is with Dr. Casey Athid, our newest assistant professor and faculty extension specialist in the Department of Crop Science at the University of Illinois Urbana-Champaign. Casey's main disciplinary focus with us is plant protection and entomology in general, but she's definitely going to be working very closely with our specialty crop audiences and small farm on some of these emerging issues. For those of you familiar with Rick Weinzer, our former colleague, this is kind of that same role that Rick played. So you can tell that we're really happy to have our entomologist back. She comes to us by way of postdoc work at Washington State University and University of Kentucky, where she also received her PhD in entomology. I would encourage you to do a scan of her cited references in Google Scholar. She's done a lot of really interesting work with molecular gut content analysis for generalist predators in conservation biological control programs. And we're really excited to have her as a resource and extension collaborator and look forward to the work her lab produces for years to come. This week, I asked her to come on to the webinar series to kind of outline her insight into biological control systems and protected culture, as well as share a little preview of her work in this space. So with that, I'll go ahead and turn it over to Casey. Thanks so much.
Well, thank you for that introduction. That was lovely. And I am really happy to be here, happy to be in Illinois. I grew up in Nebraska, and so being back in the Midwest is just exactly where I want to be. As Zach said, today I'm going to talk about biological control in greenhouses and high tunnels. And I want to start by giving you some background on the concept of biological control and the types and where it's successful. I have worked primarily in conservation biological control up till now, and I am moving into the world of augmentative biological control, and I'll go over what both of those are. And then I also want to give you this preview of high tunnel research I will be doing this summer. And I think that research is going to be, the results of that are going to be really interesting for growers. The purpose of that is actually to get you guys some research-backed recommendations for use of biological control in specifically high tunnels. And so I want to start with the definition of biological control. And it's defined as the suppression or prevention of a pest outbreak due to the purposeful manipulation of natural enemies. And these are either predators or parasitoids primarily. And I have included this slide is a picture of a bunch of biological control agents, or at least the common ones. And you can see on the left, what I have, these two are both lady beetles. You'd recognize this as the adult lady beetle. This is the immature lady beetle. They all life stages are great for controlling aphids. It's a very common biological control agent and one that I'll be talking about quite a bit through this talk. And then over here on the right, you'll see this is called the minute pirate bug. This is another really fantastic biological control agent that that's commercially available. You may recognize this guy as this fall. They definitely also bite, which is sort of a side of another thing I did not know about them, but they are great for consuming these small greenhouse and high tunnel pests. And then, because I am really interested in spiders, I included a picture of a spider down here eating a spotted cucumber beetle. Spiders are not commercially available, but they can be considered biological control agents, given that they're often in high tunnels already or in fields already, and they are pretty voracious predators. Now in the middle is a group that I'm not going to talk as much about, but I will a little bit, because my primary focus is really on these predators. But parasitoids are another big group of natural enemies, and many of them are biological control agents. So you can see here, this is actually a natural enemy of a tomato hornworm. This is one that attacks whiteflies. And this picture, this is the one that does whiteflies. This is tomato hornworm, if you can see my cursor there. And then this picture over here that looks like a bunch of seeds is actually showing you whiteflies that have been parasitized by one of these wasps, and these little black seed-like things. And I'll talk more about that a little later as well, with things you might see if you have parasitoid activity. So biological control is considered environmentally safe, and it's quite effective with a number of successes, including its use in greenhouses. And it can be considered cost effective in that it can have a great potential for a return in a small investment. The other thing, and the main thing that biological control has going for it, is it can reduce the reliance on broad spectrum chemical insecticides for control of pests. So to be more specific on sort of the objectives here, the objective of biological control is to either prevent, reduce, or delay a pest population. And I talked a lot about the predators and parasitoids on the previous slide. The one that I'm really not going to talk a lot about at all are diseases. So here in this bottom right, you'll see this is a weevil covered in a fungal disease, and you can actually purchase these diseases like the fungus spores as biological control agents as well, but I'm not really going to talk about those too much. And so one other thing I want to note going forward is I'm often going to use the word predator in this talk, and I'm primarily going to use the word predator. And that's really just a shorthand. Just keep in mind that biological control agents can be a bunch of different things. And when I say predator, you mean that to encompass sort of all of the things we would use here. But my primary focus, again, is predators and not so much the diseases and the parasitoids. So there are three broad types of biological control that I'm going to go over before I talk about the specifics of biological control in protected culture. And these are the importation biological control, conservation biological control, and augmentative biological control. And of course, as the title of this talk suggests, augmentative biological control is what be using primarily in greenhouses and high tunnels. And there are actually two types of augmentative biological control, which I'll go over as well. And that's the inundation or inoculation. So I want to start with importation biological control because this is the first type of biological control that entomologists ever used. It is also known as classical biological control. And this is where we actually introduce exotic predators to control pests. And importation biological control is often used to target an invasive species. And usually these invasive species arrive in a new area and they don't have any of their natural predators at all. And so the native predators that we have here don't usually switch to the new invasive pest, at least not in any form that really can control it. And so natural control of these pests seldom works. And of course, you guys are all probably all familiar of a bunch of these. The main one in recent years being the brown marmorated stink bug, just showing up and really not having any predators to speak of that could take care of their populations. And what we need to do here is we actually need to import the native predator of this pest to the new area to reestablish the relationship between this invasive pest and its predator. And there are many successes to this that have happened throughout the last 100 years that we've essentially been doing this. And there are some famous failures as well. One that comes to mind is a thing that the Asian lady beetle, which does do great with aphid control, but it also displaces native lady beetles. It comes into people's houses. It sours wine. It does a variety of things that aren't great. And so that would be kind of considered a failure because although it does kind of do the thing we wanted it to, it has a lot of non-target effects. And I found this graphic in a paper, and this is an infographic to show you sort of how this type of biological control works. And in this example, the invasive pest is actually a plant. And so they would go to the native range of this invasive pest and find these beetles that are eating the plant, transport them back over here. I added quarantine in here because you always have to quarantine these pests. You always have to do lots of tests to make sure they're not going to eat other things. And then you introduce them, hope that they establish, and then they spread to other plants, and that their ultimate impact is helping to control that invasive species. And with this, the idea would be that it is pretty cost effective because if you can get to the point where they're establishing spread and having impact, you're not needing to re-release things into the environment. And so this type of biological control has the capacity to be massive reward for very little input. The second type is the type that I have primarily focused on with my research up until now. And this is conservation biological control. And this is where you actually want to keep and enhance the predators that are already in the landscape. And this requires an active manipulation of the landscape or your management practices. So conservation biological control is not just the natural control by the predators in the system. You actually have to do something to help out those predators in order to be doing conservation biological control. And so some of the ways that you can do this is altering management practices, such as using things like strip cropping or changing to a polyculture. Now, of course, if you have home gardens or a lot of small growers are already in polyculture, one of the other things that people can do is use things like unmowed edges or weed strips or unsprayed edges. And what this does is this can provide your predators with food like sugar or nectar or pollen sources. And these plants can attract and retain predators to an area, even when their prey items like the pests that you want them to control are not actually available. So then when the pests arrive in the fields, the predators are there and they'll be able to start eating the pests. The picture on the right is a wildflower strip, which provides resources for these predators. And then the other thing to do is if you're wanting to keep and enhance the predators already in your landscape, you can alter the use and timing of pesticides. And the main way to do this to reduce your pesticide use. And this is something that will be important later in the talk is how pesticide use may affect some of these natural enemies. Now, of course, these pictures are, open field systems. And so there is some conservation biological control tends to be more relevant in open field systems. And then, of course, to get to the type of biological control that is more relevant in high tunnels and greenhouses is augmentative biological control. And so this is where you add predators to the environment. And these are meant to supplement any existing predators that you might have in the environment already. And you can release one species or several. And the goal of this is generally not the permanent establishment of the predators. And this is especially true if you're doing this in something like a high tunnel or a greenhouse where you have a certain time of the year where you don't have any plants in there because your predators obviously will not survive the overwintering. And so you'll have to keep releasing. And there are many commercially available predators that can be easily purchased. And then there are two types of this. And the first is this inundation, where you release large numbers of predators that you hope will overwhelm the pests and provide a quick knockdown effect. And the second type is inoculation. And this is when you would release a small number in the early season. And you hope that your predator will reproduce through the season and keep those pest numbers low for a longer time. And inundation is definitely more common for predators, just because often once we see a problem, then we really have to hit it hard with a large number of predators. And as I said, this is the type of biological control that you'll be doing in high tunnels and greenhouses. So, as I said, I've been using predator as a shorthand, but you can buy both predators and parasitoids, which are small wasps generally. And a mix of these is often a good idea in protected culture. And there are a lot of options for purchase of beneficial organisms. And this actually includes things like pollinators as well. You can purchase pollinators from a variety of vendors that aren't just honeybees. And the University of Kentucky has a list of commercially available predators and parasitoids, as well as all the vendors where you can purchase them. And it's at this website. And I'm going to actually open that up in a second and kind of go over a little bit of it for you, because I think it's a really good resource for when you're considering purchasing some of these things. And it's a good thing to look back on and see what's potentially available and all the different companies that sell them. And I also want to note that what we'll see in just a second is that there are really two types of these natural enemies. There are things that are specialists that you're really going to purchase if you have one specific pest problem. And then there are things that are considered generalists, and those are predators that kind of eat anything. And so if you have a variety of pests, you may want to be looking at more of these generalists, depending on what you've got as far as that goes. So let's see. Let me switch over. Okay, so you guys can see that this is and you can also download this as a PDF if you'd like. So this is vendors of beneficial organisms in North America. And I'd like to point out a couple of things. There's a bunch of good information here at the top, and then some other websites to go to read more about it. But I want to start here, and I want to point you to these beneficial organisms currently available. Now, I should note that this fact sheet was made, I think, 2 years ago or 3. And so when you see this exhaustive list of natural enemies that you can purchase, some of these may not be available at any given time. But so you can see that the way that she's divided this up is what type of control the natural enemy does. So here up top, we have Aphid Control, and these are all wasps and one fly that only control aphids, and so on. So Beetle Control, got a lot of things that control flies. And then here, this list of general predators and parasites. So these are things that you can purchase that would attack pretty much anything. And as you can see from this list, they've all got these abbreviations in front of the species that that you can order. And so you can see on this list, there's a lot of stuff that is available for control of different pests. So here's mite control. And then you can see here, there, like I said, they also have pollinators available. And then there's a bunch of things for weed control. And then as we move down, you'll see these retail and wholesale suppliers. And the way that these are divided is they're in alphabetical order. And then underneath, she lists the various organisms they have for sale. And you can see they're done by those abbreviations. And so you will have to sort of look at what's available and go back up on the list and see if those would be appropriate for the pest issue that you have. I'd like to point out right here, Arbico is the one that I generally use and is used often by researchers. And there's another one down the list called BioBest. And as you can see with Arbico, they have a lot of organisms available that you can purchase. So they're a pretty big company for that. And so that's a good one to keep in mind. But I've put this link, obviously it's right there on the slide. And so feel free to peruse that website. But it does have, like, I mean, as I'm scrolling down, there's a lot of suppliers for these natural enemies. So let me go back to my slides. Okay. So moving along, there will be a lot of things that you'll have to keep in mind if you're looking to purchase biological control agents, because of course it's not as simple as just buying a chemical insecticide. Because these are living organisms that you're using, their quality is going to be affected by the shipping and storage. The predators that you order may need to acclimate to the environmental conditions that you're going to release them in before you release them. And one of the things you're gonna wanna do if you're doing this for the first time is you're gonna wanna contact the vendors. Now, some vendors have really fantastic websites that have a lot of information on how they store their insects, where they come from, that sort of thing. But you can always, and you should, contact the vendors and ask them specific questions before you purchase these insects. Because you really want to make sure that when you purchase this, it is doing what you hope it will do, and that it's not getting damaged in shipping or being released in a way that means that it won't be as effective against the pests that you're hoping it will control. So some of the specific questions you want to ask is, you want to ask these vendors if the predators are shipping directly from the insectary. So some of these vendors actually raise the insects themselves, some do not. And so they may be in storage someplace. And you're going to want to know kind of where they come from and how long they're going to be in storage. And so that's the second question, is how long are they stored before they are shipped? And you won't ask how they're shipped, just get an idea of what kind of conditions they're going to be in. So for example, if you're ordering some really small predators to attack things like thrips or mites, they will often come in a big container that looks like it's full of sawdust. And when you open it up, it's like there's nothing in there. And then you'll see that the sawdust sort of seems to be moving. And that's just information, so you won't be surprised when you get your natural enemies. Some predators are shipped as eggs, some are shipped as adults. That's all information that you'll want to have ahead of time before you order them. And then one of the big things is, what will they look like when you get them, and how can you tell as the buyer that you have high quality insects that have been shipped? And then I suppose I put in bold, and I feel that this is probably the most important question to ask, is what are the best practices for release of this predator? So different predators will you'll want to release them at different times of the day. That's kind of important, whether they need to be released at night or in the morning or in the middle of the day or that sort of thing. Because if you release them at the wrong time, they may not get established the way that we would like them to, so that they can be doing that pest control right away. And as far as success goes, one of the main ways to make biological control successful is to start when your pest numbers are low. If your numbers are already pretty out of control, you can still use biological control, but you're going to need a lot more insects to do it. And it will be a lot harder to control than if you just started when the numbers are low. And so the key to using augmented biological control is prevention. Biological control agents often work slowly. They're very different from an insecticidal spray. And so to decrease your pest numbers, you want to cause mortality early against your target species. And you may be able to prevent or delay any, or prevent injury and damage and delay your pest numbers past when they actually can do damage to your crop. And so this requires monitoring and intervention. And biological control is useful against sort of regular and predictable pests. And it should also be noted that predators can have different levels of effectiveness depending on the plant. And so that's also something that you may want to ask a vendor of a biological control agent if there are certain plants it really doesn't work very well on. So the first step here is to actually identify your pest and determine its population size. And to detect early infestations, a crop scouting program that includes both yellow sticky cards, which you can see there on the right, and visual inspection is critical. And scouting should be done at least once a week, and more often once you actually detect an infestation. And that is also for monitoring the efficacy of your control measures. A hand lens is really useful in this sort of monitoring to detect live pests as well as signs of their activity, things like cast skins or honeydew, that sort of thing. And especially considering most of the pests that you're going to be concerned with that you can use biological control on in high tunnels and greenhouses are very small. So the next part of this talk is just going to be going over the main greenhouse and high tunnel pests you're probably going to encounter. When you are trying to scout and monitor for pest problems, on top of looking for the actual pests themselves, looking at some of the damage seeing the damage that they're causing to your plants, maybe another good cue, because all of these pests are very, very small. And you'll find these pests in open fields too, but they do tend to be worse in protected culture. We tend to call these greenhouse pests. And on the upper left here, these are spider mites. And the center, top, and right are aphids. And then the bottom left are whiteflies, and then the bottom right are thrips. One thing to note, if you haven't dealt a lot with thrips, this is just a silly thing, but one thrips is a thrips, 100 thrips are thrips. I don't know why, that's the name of it. Anyway, so like I said, since scouting and monitoring are important in these, I'm going to go over to these groups and talk about the damage you might see. So be on the lookout for them. And these pests are fantastic candidates for using augmentative biocontrol. There are broad spectrum insecticides that are labeled for use on these pests, but more often than not, when you try to spray for these, you end up with secondary pest outbreaks. These are really common pests that if people try to spray for, they tend to make the problem worse than it was without spraying. And especially in a high tunnel situation where you would expect to have some natural enemies present, if you spray broad spectrum insecticides in the hope of getting rid of these pests, you're often just going to kill off the natural enemies that are already there, which is usually the reason that we see those secondary pest outbreaks. So I'm going to start with aphids, which people are probably generally familiar with. And they can have up to 30 generations per year. And some of these life cycles are as short as 12 days. And if they're not controlled, they can experience exponential growth. Aphids, and actually in this middle picture here of these red aphids, you can see they often give live birth. And often when they're giving birth, the offspring are also already pregnant, which is called telescoping generations. Aphids are super weird. And you can see that the numbers can be very, very high on plants. Over here on the right is corn during anthesis, and you can see all the little blue-green spots all over here. It's just a crazy corn leaf aphid infestation there. And so these pests can get out of control kind of quickly. And they do tend to be worse in high tunnels and greenhouses. And they're particularly destructive pests. because they feed on, they're foliage feeders and they spread lots of plant viruses. And the feeding can cause distortion of new growth, which you can see in this picture of some Brussels sprouts. And they produce honeydew, which you can see down here in this photo, that causes sooty mold. And the sooty mold can also grow on the fruit, and that can cause yield losses. And so there's a variety of options for aphid control for predators that are available. And there is in this picture, you can see this top picture, this yellow slug looking thing. This is actually a fly larva that is a specialist on aphids. And then there's lady beetles right here in the middle. These two guys are some commercially available lady beetles. And then there's also lacewing larvae. Now, I would recommend the lady beetles for control. You'll see lacewing larvae a lot. You'll see them as most of the vendors will sell them. They usually sell them as eggs. I'm not sure how effective they actually are in greenhouses and high tunnels for aphid control. Because they're sold as eggs, they tend to be sort of hard to deal with. And as adults, they're actually not predators. So the nice thing about lady beetles is lady beetles are predators as larvae and adults. So you get kind of that longer control with them if you can get them to reproduce in your greenhouse or your high tunnel. And so that's really the group that I recommend for purchase for aphid control. And on that note, I'm going to give a little bit more recommendation on those. So if infestations are high, your release rates need to be really high. And depending on the time of the year, it can sometimes be difficult to obtain lady beetles because they are really popular biological control agents. So just keep that in mind. When your shipments arrive with lady beetles, it is useful to let them rest in the box in an area with an appropriate temperature. So inside your greenhouse area or inside your high tunnel is fine. And you want to release them just before, either in a greenhouse, the lights are turned off, or just before dark in the high tunnel. This will allow them to get over the stress of shipment and settle down a bit. So lady beetles have this annoying behavior where they will try to escape and they will go to the edges of your facility. And in a high tunnel, that might result in them actually leaving the high tunnel. And so you really want to be careful. This is one of the reasons why I recommend talking to vendors before you purchase something, because with lady beetles, they will try to escape if they are not properly acclimatized. And in addition to predators, there are several aphid parasitoids for sale that are also really effective. And so if you wanted to mix parasitoids with predators, you could potentially do that with the knowledge that those predators may eat the parasitized aphids. So these are small wasps that lay an egg inside an aphid, and then the aphid dies and the wasp emerges from it. So in this picture on the right, it's a little bit grainy, but you can see this wasp attacking this aphid. So I've included these photos because it is likely that if you have an aphid infestation, especially in a high tunnel, you already have parasitoids within that infestation. So the parasitized aphids are called mummies, and you can see them here. They look bloated and they get brown, and they're actually pretty hard. They get sort of sclerotized. And if you see this among your aphids, that's a good sign. That's shown that there's some natural control happening. And of course, this is often what gets disrupted when we spray broad spectrum insecticides for aphid control is these guys die much. These wasps are really sensitive to that. And so this is something just to be on the lookout in your high tunnels to see if you're getting some natural control of the aphids. And I want to show you one more picture showing biological control in action of aphids. So this one, again, you can see lots of mummies on this leaf, but I wanted to point out down here, these yellow things right here are lady beetle eggs. And so lady beetles, being voracious aphid predators, will often lay their eggs amongst lady beetles or amongst aphids. So when the larvae hatch, they're right where their food is. And so again, you would see this and think, oh, there's a lot of good natural control happening. And really, a lot of parasitized aphids there, which is fantastic. So mites are our next group, and they have 6 to 8 generations per year often, but sometimes their generation time can be as short as 8 days. And they're capable of exponential growth, just like aphids. And they generally cause issues to the foliage. And one of the ways you can tell that you have spider mites often is if you see this webbing, because like the name implies, they have this silk. And if it's not just a spider web, that's a pretty good indication you have a spider mite infestation. And then, they tend to do this yellow and white stippling, and you get reddish or pale discoloration to leaves. Your plants can lose vigor. You can get premature leaf drop. And then there can also be damage to fruit. And I've included this picture of a pepper here that's pretty extreme. This is extreme spider mite damage to this pepper. And they can be difficult to control. And so, again, there are lots of options for mite control, and predatory mites are the main thing that you would want to purchase for this. These are commercially available. They're pretty effective. There are some other predators. I have some pictures here. This is actually it's a tiny lady beetle. And then this little guy down here is a fly larvae, and that's also a pretty voracious mite eater. So again, you have lots of options that are available, and these are effective mite predators. And the next group that you would commonly find in these areas are the thrips. And again, they can have really short life cycles, as little as 2 weeks. And the females actually lay their eggs on or into leaves, buds, fruit, and other plant locations. And then, like aphids, thrips can transmit plant diseases, and they can cause damage to both foliage and fruit. And so I've got a lot of pictures here. Down here is a pretty cool picture of a thrips there in the bottom left. And here on this pepper flower, you'll see a bunch of they're really small, so you can see the thrips. These are actually pretty big for thrips on a pepper flower. But here in this bottom left, you can see that is a hole from actually a thrips laying her eggs in this tomato. Up here on this pepper is a bunch of discoloration from thrips feeding. And then you can see this discoloration in these tomatoes as well from thrips feeding. So it can definitely result in quite a bit of yield loss. So thrips control is a lot like mite control, in that you buy predatory mites. And so you can actually buy, I believe it's this one, which is the same for both. And then the minute pirate bug, this guy over here, is a pretty effective thrips predator as well. And then something that like I said, I haven't talked about much, but I did want to mention here, there's a nematode, these worms, that is available for thrips control, which is pretty effective as well, that you can purchase. And then the last group of these greenhouse and high tunnel pests that I want to discuss are just the whiteflies. And you'll note a theme here that, much like these other three, the populations of whiteflies can build up really, really quickly. And they lay their eggs on the underside of leaves. And they have these life stages that called the crawler, which is the one that's actually moving around. And then after that, they lay down on the plant and they're really immobile, much like scales are, until they're adults again. And you can see here in this right picture is some discoloration and damage from whitefly feeding. They also produce honeydew like aphids do, so their feeding can result in sooty mold. And whiteflies can be controlled by this species of predatory mite as well. So this is a really good one to purchase if you have multiple small, if you have mites and thrips and whiteflies, you may want to just purchase this predatory mite because it might, in large numbers, take care of some of your other problems. And then there's a tiny beetle that you can purchase, and then this little true bug that are pretty effective whitefly predators. However, with whiteflies, really, the parasitoids are going to be where you want to go for this. And specifically, there are two species, these two, that you actually usually purchase as a set, because they'll attack the two main whitefly species that you'll find in greenhouses and high tunnels. And I wanted to point out once again, I showed this picture earlier, but I just wanted to sort of point it out. Much like aphids, with whiteflies, you can recognize when whiteflies have been parasitized. So the whiteflies, when they're not moving around, they're these little flat things right here, little kind of yellowy guys. When they're parasitized, I've got these orange arrows. When they're parasitized, they're black. So when you look at that, it looks like a bunch of black seeds on the bottom, and that is showing you that there's parasitoid activity. And so if you have natural control in the area, you would expect to find those if you had whiteflies in an open field. So for biological control in protected culture, there is lots of evidence that biological control in greenhouses is quite effective. The pest issues in greenhouses are pretty predictable, mostly, based on a bunch of factors. For one, greenhouses are completely enclosed and don't have a lot of pest immigration from the surrounding landscape. And there are a number of pest groups that are associated strongly with greenhouses. And greenhouses are also generally climate controlled. And because the predators cannot easily escape, as long as the environment is hospitable and they don't die, they will happily eat the pests in the greenhouse. And this is especially true if you've properly identified your pests and the predators and parasitoids are tailored to your pest control needs. High tunnels, on the other hand, are permeable, and there is generally less temperature control. And this is because they are really a mix of greenhouse and open field conditions. And as a result, they can have, both greenhouse and open field pests, which really complicates the pest control situation. And the effectiveness of predators in high tunnel systems is not fully known. And research on the subject is kind of rare. There is some, but it's really just getting started. And it really shows mixed results because I think there's still a lot of questions. And one of the ones that comes up often is like, when's the best time to release these predators so that, I mentioned earlier that lady beetles tend to want to escape if you release them at the wrong time. So what is the right time in a high tunnel versus the right time in a greenhouse? And so as a result of this, I really wanted to look into some of these high tunnel questions. And so I have a project that I'll be doing next summer that I wanted to just talk a little about and introduce you to. So this is a project that's actually a collaboration with extension folks. Down at Dixon Springs and then up on campus as well. And the idea of it actually kind of came from Bronwyn Allie, because she saw a need for this. And so we kind of ran with it as a project to work with next summer. And so we're going to be investigating biological control of tomato and pepper pests in high tunnels. And because they are expected to have both greenhouse pests, such as, aphids and thrips that I've been talking about, and open field pests, such as caterpillars like the tomato hornworm or stink bugs, we want to look at how we control those different types of pests. And as I mentioned, although their use is expanding, high tunnel use, there are really very few research-backed insect control recommendations that we can make to growers currently. And so what I'm going to do is I'm going to release commercially available generalist predators, they're going to consist of lady beetles and minute pirate bugs. And if I can get them, there is a predatory stink bug I'd like to release as well, but they're not always available for purchase. And what I'm going to do is I'm going to release them, and then I'm going to survey the ones I've released to see how they're established, and then also the resident predators as well, which will include other lady beetle species and then spiders and other predatory bugs. I'm then going to employ something called molecular gut content analysis, which I'll talk just a little bit about, to determine which predators are actually consuming the pests. And I'm going to be doing pest sampling and monitoring to see if our releases are actually impacting the pest populations in the high tunnels. And I'm kind of expecting that maybe these released natural enemies will really be having more of an effect on the greenhouse pests, and maybe the resident natural enemies might be affecting these more open field pests. But I do want to just talk a little bit about how we figure out what the predators are eating. And so one of the main questions of this project is which ones are doing the pest control. And if it's not the commercially released predators, maybe we can just use conservation biological control in high tunnels, like planting flowers in the outer rows to draw in more resident natural enemies. And so we use molecular gut content analysis, which is just a fancy way of saying we just use DNA to determine what the predator ate. And so I have this little drawing of a spider, and in this scenario, let's say this spider ate this stink bug nymph. And of course, you can't know that by looking at it. And so I want to be able to confirm that because maybe I'm interested in stink bug control, which would be true for this project as well. And so I design, I'm going to call it a marker that detects the DNA of this stink bug. And so the way that we do this is we take our spider with its last meal in its guts, and we smash up the whole thing, and we put it in a tube with some chemicals, allowing us to free the DNA from the spider in its guts. In this picture, what I'm showing you is that the spider DNA is in red and the gut DNA is in black. And so we get a lot more DNA from the predator than we do of the actual thing it ate. And that's where this marker that I made for stink bugs come in. So we use this, and it makes just a bunch of copies of the stink bug DNA. And then we use this thing over here, this visualization, to see it. In this case, you see this fluorescent thing by my cursor. If that band shows up, that means there's stink bug DNA. If it doesn't, like here, it means there's not. And so one other thing we can do with this technique, which will be useful for pest control in high tunnels because there's often more than one pest, is we can actually detect if our predator eats more than one thing. And so I will be doing that this summer. I will be testing these predators for eating whatever the pest issues in those high tunnels are, whether it's aphids, whether it's mites, whether it's whiteflies, stink bugs, or all of them. I'll be testing these predators for all of those things using individual markers. And so, you know, as I mentioned, sort of these expected results for this is that the commercial biological control agents I would expect to really be feeding on the greenhouse pests and impacting their populations. And of course, we're purchasing ones that are tailored for greenhouse pests. I would expect the resident predators, things like spiders and that sort of thing, to be consuming some of those open field pests, such as maybe some of the caterpillar issues. And what we're going to want to ultimately do is create these research-backed recommendations for high tunnel growers that are looking to use biological control, whether it ends up being augmentative biological control or a conservation biological control technique to bring in the resident natural enemies. And so I'm hoping by the end of next summer, I'll have at least some preliminary things to share with you to really start making those recommendations for high tunnel insect control. And with that, I do have this QR code for the survey. And I do have one more slide. I didn't put this on my exact last slide. I do have one more slide with my contact information that I can put up in a bit. But that is all I have. If anybody has any questions or would like to discuss anything further, I will take all of that now.
Great. Well, thank you so much, Casey. This was very useful. You can go ahead and put up your contact info slide if you want.
Okay.
I have the evaluation link here in the chat box as well. So
Okay.
click on the hyperlink. So a couple questions that came in. I have a couple prompts, and I'm sure a couple questions will come in. But we'll start with the ones that I do have written down. There was a question that came in relating to particular plants you can grow that will naturally attract the predators for the common greenhouse pest. So this nod to your conversation about conservation, biological control, farmscaping. I put a link in the box. I don't know if you're familiar with the Xerxes Society. They have a wonderful farming with native beneficials book. That's a practical manual that kind of outlines some of these, clustered plant species as well as techniques. Are there any particular recommendations around plant selection for conservation biocontrol you can make?
Not necessarily, because what you really want is you want things that have like a lot of nectar. And you want, if you can, what we think works pretty well is things like a wildflower mix, where you might have flowers at different times, and you might have, because individual species might be looking for a different type of nectar, a different type of flower, or the pollen is available at different times. And so really, if you're doing like a flower strip, a mix of flowers is always a good way to go. I think there's a lot of recommendations on what particular species to use. But, one of the things we like to try to get back to is things that are maybe more natives, as opposed to some of the non-native species that we often plant in landscapes that look really pretty. And so those native wildflower packs are probably a good way to go, because then you get flowers at different times and you might get your nectar sources at different times. I don't have any specific recommendations, but I could go and see if there are some particular flowers or plants that have been really well studied on that besides the wildflower mixes, and get back to you for sure.
No, that's great. That's great advice. One thing I'm knowing, maybe you can speak to this, and if you look at references when it talks about farmscaping for conservation biological control, is it's one thing to to purchase these seeds, these plants, these mixes, and oftentimes farmers might sort of have a designated area for this, and they pay very little attention or little effort in getting those plantings established. In other words, just like we talked about with cover crops, that you should treat these plantings like you would your cash crops. Can you add to that? Have you noticed that on farms that you've worked with, where it's kind of, an afterthought to do farmscaping for conservation biological control? Is it important to pay attention to establishing those species to have success with that?
So it's interesting, because I can't speak to for sure about whether or not you ignore it or, just sort of let things go. But one thing that I saw actually like 2 days ago, so there's some researchers in Montana that are working on getting wildflower strips to be economically beneficial to the grower. So to really make this work economically. And one thing that they found was that if you are a grower and you're actually harvesting seeds from your wildflower strips and selling them, you make more money. Like, that makes the economics of the system work much better. And so in that case, you would want to be putting at least some level of time into making sure that those wildflower strips are establishing, if you're then harvesting seeds from them. Now, this was just one pilot study. It's in Montana, and it's really the first one I've seen that looking at the economics of planting wildflower strips outside of just their benefit to biological control agents. But I thought it was really interesting. And definitely makes the case for spending a little bit more time. But I will say that weed strips tend to work just as well as wildflowers, except that, of course, as a grower, you don't want a weed seed bank, which is why you want to plant something that give wildflowers or whatever else. But weed strips often will have a good mix of plants that are really good for natural enemies, for resources they're looking for. But of course, most growers are pretty reluctant to say, like, I'm just going to let these weeds grow up next to my fields, especially organic growers who don't have really another option for weed control. But yeah, you can some of those things you can kind of sort of just let go, and those natural enemies will show up to a lot of plants that might be kind of surprising.
Great. Great insight. So another question that came in was specific around fungus gnats and control. I know these are often a problem, particularly with container production
Mhm.
greenhouses. Do you have anything off the top related to fungus gnats? I mean, that might be more of a deep dive question.
I don't, but I know that there are, I shouldn't say I know, I'm pretty sure there are some pretty effective parasitoids, I want to say, of those guys, but I would have to look that one up. Now, I think there were some fungus gnat control on that web page I put. I think
Okay.
there were some on that, but I can definitely look that up because that was one pest group that I did neglect to talk about today. But yeah, they're definitely for container, and actually just like in greenhouses too, you can have, even outside of the container, like within the greenhouse, you can have pretty bad fungus gnat issues. One thing to do about fungus gnats often is if you can cover the soil really well. So one of the ways that we dealt with fungus gnats in containers was actually putting a layer of plaster over the soil. So just pouring plaster over it, because plaster is permeable, so you can still put the water in there, and that will take care of the fungus gnats. But that's pretty labor intensive. That would really be if you had, just a small number of containers. But that when you're doing research in greenhouses, often you'll do that because you don't want the fungus gnats there if they're not what you were focusing on, because they can confound the research that you're trying to do. And so that is one, non-insecticidal, non-biocontrol way of doing it, is to really get that soil as covered as you can and kind of suffocate those gnats.
Excellent. So a question came in, and this is kind of a larger, particularly you mentioned like the stink bug and some of these larger insect pests. This, cucumber beetles, and bacterial wilt transmission in the field, but also in the high tunnels can be an issue. You mentioned you did a great job of sort of comparing, really controlled environment greenhouses versus relatively more open high tunnels. My recommendation to this comment was greenhouse screening, which is something we try to use. We try to actually screen our high tunnels. Can you speak to that at all in terms of the effectiveness of insect screening or greenhouse specific screening for some of these more sort of high tunnel type protected culture?
Yeah. You can keep, if you use screening that is not, it doesn't even have to be all that small, but you'll keep things like squash bugs and stink bugs and cucumber beetles out with pretty, I shouldn't say large, but relatively large screening. Of course, you're not going to keep things like aphids out, but the other thing you won't keep out is small parasitoids will be able to get through. And so it's a good way. I worked in cucurbit row cover systems, and we went from the regular fabric, like spun bonded remay fabric covers, to basically a netting that had much larger holes in it, and we were really able to keep out those large pests pretty easily. We had still things like aphids and stuff, but it's really hard to keep things like thrips and aphids out with screening just because they're so small. But your big things, I think those will be quite effective to just screen things off if those are the ones you're worried about, for sure.
And there's a balance with temperature management with those as well.
There
is
is.
a screen that is small enough for aphids, but then it curtails the air, things like that.
Yeah, no, and I think that was a calculus we made too with the row cover, because if you get it too small, one of the things with the remay was that it got too hot under there. And so if we made that netting too small, we weren't really fixing the it's getting too hot in here problem. So yeah, no, that's absolutely true. Like that's another calculus to be made for sure.
So we have a few more questions coming. I just want to remind everyone we're coming up to the 1 o'clock hour, so we'll continue with this Q&A with Dr. Athey, and if you need to hop off, by all means, but we'll go ahead and keep this recorded so you can get the link a little bit later. So a couple of questions. This relates to ladybugs or parasitic wasps for aphids in particular with open garden production, I think is the question. And I think you kind of talked a little bit about the difference in effectiveness with augmented biological control and in a controlled setting versus open field. Do you want to maybe re-emphasize that or comment about that?
Yeah. So, you're going to expect to see, and I would be surprised if you didn't have, when you have an open field, if you didn't have lady beetles and parasitoids already. And so really that's an area where you want to think about what conservation biological control might do for you. The issue with releasing something like a lady beetle in an open field is it is going to fly away. It might stay for a while, but natural enemies are not inexpensive. It's a good way to go, but it's not as if they're really cheap. And so I think recommendations for releasing something in an augmented biological control in an open field is harder for me to recommend. Now this might be an area where something like those lacewing larvae could be more effective, depending. I feel like lacewing larvae are often recommended, but because again, you get them as eggs and you put them out, and it's really hard to know if they're doing what you hope they would do, but their effectiveness is when they're larvae and they don't fly away. So that's a place where open fields might be sort of the area that you want to do something like that. Or like that fly larva, that little slug looking thing I showed in the Aphid Control slide. That one is also not going to escape because it doesn't have wings. You put it someplace and it's likely going to stay there. And so really for open field conditions, it might be more of those types of biological control agents that you want to be focused on versus something like lady beetles that are fantastic, but they will just go wherever they want.
Okay. Excellent. So I mentioned the Xerxes Society reference for Farmscaping, conservation biological control. Do you have any other good resources off the top of your head for things like planting flower strips, pollinator strips, things like that? Are there any popular ones that you can think of? That was one question.
So there's a few. Again, University of Kentucky has a lot of really good fact sheets on biological control because they've got a few people who do quite a lot of research on that. But off the top of my head, as far as Augmentative goes, that website that I showed you does have a few more links on biological control from there as well. And I probably should have some off the top of my head, but I don't really have any other ones for sure that really talk about the conservation biological control techniques.
Yeah, and if you think of some, we can pull them together when we send out the links, or we can always put them in the comment section of the YouTube channel as well.
I'll find some links that are good to be able to share with people.
Okay. There's another question coming in here about sweet sorghum as a trap crop. Mentioned sweet sorghum near tomatoes with hornworms, grasshoppers. I know that there's some work with brown marmorated stink bug with sorghum as a trap crop. Do you have any experience or knowledge about sweet sorghum as a trap?
Not as a trap, because a lot of the work that was done on sweet sorghum at the places I've been has been on the aphids that infest sorghum, as opposed to using it as a trap crop. One of the issues with trap cropping generally, so it's not that it's a bad idea to use, but often depending on, unless what you're doing is trap cropping and then specifically spraying there. So there's a fair amount of research of like attracting in stink bugs to an area and then only spraying that area. So you're then hitting that with insecticides. Because otherwise, sometimes if you're just using it and hoping like they'll stay there, of course they don't. And so that can sometimes, there is occasionally some evidence to suggest that a trap crop could potentially bring more things into the area than would have been otherwise. But I don't know specifically about sweet sorghum, and let me also look that up and see what the, if there's some good, I'm sure there's a lot of research done on that I'm not aware of myself, and see if there's some good recommendations, because it's possible they're just planting it and then once, if you're spraying then in that one area, that's probably a pretty good way to go.
Right. So there was a comment on here related to the fungus gnat conversation about maybe mulches possibly working as a barrier. Do you have any experience with that?
I don't, and I would say that as a container, so like as far as mulch goes, if you mean by like the plastic mulching that goes over the top, the reason that we used plaster is because they couldn't possibly get in or out. You know, unless you're using something that is really, really tight, you're still probably going to get some of those fungus gnats. And it may be a better way to prevent it, because again, if you can cover any of that, it's a good way to go. But I'm not sure as far as container planting goes, how effective a mulch would be.
Okay, so just a couple more things here. One, so the cucumber beetle thing popped up again with the reference about how, yes, cucumber beetles, if you already have a problem with them in a tunnel, say a non-movable tunnel, and they're overwintering in the soil, screening wouldn't be effective. Are there good predators or parasitoids of cucumber beetles? I've never heard of that as a control option. Or
no, not really. And most of the problem with cucumber beetles and things like them is when you have a larvae in the soil, we as entomologists kind of don't do it. It's hard to do soil research. Kind of, I guess, would be the easiest way to put that. Probably if you were looking to do soil-borne control of cucumber beetles, some of the nematodes are probably where you would go as far as that goes. However, I don't believe there's much research on that. You know, the adult beetles they're not huge, but one of the things about biological control agents is they're often targeting smaller things. And so you will see spiders occasionally taking down cucumber beetles, but not in any level that's really having really any effect on the populations. And so you could maybe use nematodes. One of the things to do, even though, you know, in a high tunnel, if you suspect that you have a lot of overwintering beetles in your soil, you can kind of try to like turn the soil up enough and really churn it enough so maybe you're breaking them up in the soil as they're overwintering. Because, and again, nematodes might be a way to go, but there's just not a lot of options for control of those sorts of pests outside of insecticide use at this point. And even the research I did, we did lots and lots of predators. I collected lots of little spiders that actually make their webs near the ground. We collected wolf spiders that run across the ground. And we thought maybe some of these small spiders that do their webs by the ground will go into cracks in the ground when it gets warm and things are too hot. And so we thought maybe they could be encountering the cucumber beetle larvae in the soil and maybe eating them. And we did not find any evidence that really anything we were collecting was eating cucumbers. Aside from like two things, maybe.
I've always been curious about the nematode for larvae in the soil, if there was any efficacy studies related to that. All right, so I don't want to extend this too longer. We could have a long conversation here. I did have one closing sort of comment or question for you related to sort of economic threshold levels for pests in a tunnel, for instance, because it sort of occurs to me that with some of these things like whiteflies and aphids, that, you know, above a certain level or below a certain level, it's, you know, you can maintain, you know, or allow for a certain amount of aphids, let's say, in a given tunnel. So in the case of using augmented biological control, is there any sort of tag along with the research into this field where it says, you know, you can use this up to a certain point where you drop below maybe an economic damage threshold for a crop, and that's good enough? Because obviously this is a proactive approach. This isn't a reactive.
It is,
solution.
So I would say with aphids, whiteflies, thrips, all of those things, the key, and this is absolutely true, is once you see them, react, do it right. It's the early season thing, because the thing about their populations is they can go up so fast that if you can really knock them down early, oftentimes, depending on the plant, the effect on the plant, like if you get an aphid outbreak really late in the season in some area, maybe, you know, you're not going to stunt your plant growth, and, you know, maybe you can deal with that a little better. But if you can really knock down the populations early, especially in something like a greenhouse or even a high tunnel, even though you're going to get some immigration from outside, that's really what you want to do. Once you see that there are lots of aphids, you could still use biological control. You just have to release so many more. And so it's hard. I didn't put any really economic thresholds because, of course, different plants will be different species will be different. And as specialty crops, it's all over the board. But really, if you start to see them, you really want to control for them with your biological control agents, because they can knock them down early in the season and then maybe you don't have any problems throughout the rest of the year. You still want to continue to monitor, but one of the things that, at least in the research that I've done so far, is that those are the types of pests that if you can control them early, they are not a problem. If you can really get them controlled. And they're the ones where if you really pinpoint that early season predation, you can really prevent any of the issues that will come later in the season. And if you miss that window, you tend to get outbreaks just because they reproduce so fast. And like I said, they give birth to young that are already pregnant with the next generation. So, I mean, aphids are just crazy. Mm-hmm. They're a very weird creature.
Excellent. Well, I think because of time, I think that'll be a good place for us to stop. I want to thank our presenter, Casey, one more time for this fabulous presentation, sharing her insights into augmentative biological control. Definitely stay tuned as we have, these mini demonstrations and projects set up with some of Casey's research throughout the state with Extension. Definitely check out the Illinois Fruit and Veg newsletter that Nathan referenced last week for updates. That would be a good place where we might highlight some of these research updates. And I'd like to thank everyone for joining us today. I hope you got something out of this that can help you plan for the season. The high tunnel season really is kind of beginning again. I've already getting my first plantings in the high tunnel, so we should start thinking about getting our plans together if we're going to use some of these biocontrol agents. So definitely look for a link to this for the YouTube video. And remember, definitely fill out that evaluation link that I have up further. We really want your feedback. And so there are some questions in there that might help Dr. Athey out in some of her research. So with that, I'm going to go ahead and stop the recording, and we really appreciate everyone hanging around for a little bit today, and we'll see everyone next week.
Thanks everybody for coming.